Hello.
Hi, how are you, Yo?
Hi. Hi, nice to meet you, Katarina. I'm good.
Happy
And you?
Monday. I'm good, thank you.
Yeah.
Yeah, I'm looking forward to this talk. So,
Ah, thank you so much.
mhm, yeah,
Is this officially start or we wait 10 more minutes?
yeah, we have like 9 minutes, so yeah,
Okay.
we will. I'm just adding the topics and then the paper link,
Thank you.
and then we have a few minutes. I hope you had a good weekend.
It's Germany is quite cold. I got a little bit cold, so later if I'm sneezing or coughing, I hope it's all right.
Yeah. Oh, Thomas is here. Hi, Thomas.
Ah,
You wanna join? I'm inviting you
hello.
to speak in case you would like to do. Yeah, here it's cold too now. I mean, it's still very mild for this time of the year. At least here in New York, it's very mild. But yeah, it's
I
still.
hope it's not COVID. So far, it's not COVID.
Oh, good. I'm glad.
Mm.
Yeah, I had a sore throat too yesterday
Oh,
and
no.
felt. But so far it's also not COVID. So let's fingers crossed it stays that way. Is there a lot of COVID right now in Germany? I don't know. Right now the
I
numbers.
mean, last week my best friend just got, so I'm a bit worried.
Oh yeah, I see. Yeah, yeah. There's this new version, right?
Yes.
That is more aggressive. I mean, the thing is, how many more like versions can there be until the virus
Mm.
is kind of becoming weaker, like mutations. I'm not
You
sure.
never know. Yeah, you never know. It's like takes forever, never end.
Yeah, exactly. Will stay with us.
Mm.
I agree. So I'm inviting people in,
Hmm.
and then I will say on Twitter that we're about to start. And yeah, there's never a time that works for everyone.
Yeah, sure.
So that's why we record the rooms.
Hmm.
Yeah, but it will. Yeah, the people are really happy that we record these rooms because, yeah, they like it would be just sad if they couldn't listen to it later on in the day. Hi everyone, we will start in around 5 minutes. Thank you for coming. We will start by introducing our guest speaker, and then she will be talking about her really interesting research. I think it's the theories of how inheritance works is changing, which people didn't think it would change, I guess, like with everything. So
Yeah,
I
it
think
is a
that's
surprise.
exactly, I think that's really interesting. So how theories are changing with new discoveries. So it's very exciting to talk about these things, I think.
I'm not very familiar with Clubhouse. I have never give a talk at here, so looks quite interesting.
Yeah, so yeah, you can relax. It's pretty not formal format, and people, a lot of other rooms and clubs work more that people just talk about something. And a lot of people don't even know what they are talking about. So, but I can, if you're interested in going to more rooms and clubs, I can give you recommendations which ones
Oh, that's great.
are good.
That's great.
And one club that is good for like news related to new technology development and policies around it. If you're also interested in something like that, they moved to Twitter Spaces, so it's tech news around the world. It's pretty good.
Tech news.
And but here there are good clubs. Another great club is Quantum Photonics. Then there's the Future Brain. And there is a few neuroscience rooms and clubs with him. He's a neuroscience PhD student at MIT. He has pretty interesting rooms related to neuroscience. So yeah, I think the interesting thing on Clubhouse is that you will have people from different backgrounds coming together and discussing a topic, and the discussions are just different and sometimes really interesting because it's people that usually wouldn't meet at the conference or something, because they're from different fields and discussing a topic. I think that's pretty interesting. So, and of course during COVID this app was very popular because
you don't have
everyone
to in
was
person.
at home.
The meeting was completely impossible.
Exactly. So a lot of people that were stuck at home would be here all day.
Oh, I see.
And chatting with people. So yeah, it
It's
was
like in US, the COVID is almost there is no regulations for COVID, right? In US.
Yeah, so it depends which states and places you are. So the New York City school district still has COVID regulation in place. So only people with vaccine can get into the building, and so on. But other places have no regulation anymore. So yeah, at the doctor's offices here in New York City, and you have to wear a mask, and so it really depends on where you are and where you want to go. Restaurants and so on, they don't ask for vaccines anymore for the card. Yeah, it's more official buildings and doctors' offices.
I think in Germany it's also quite released now. So, but because I'm working in a hospital, so they still require the mask, vaccination, and everything, and we still cannot have like in-person lab meeting. I mean, now our lab meeting are mainly online. So,
Oh, okay. So we never have lab meetings. Oh, I didn't know. Yeah, my brother, he's in Essen working in a
oh, I
hospital.
see.
Yeah,
Our lab meeting is like that.
I didn't ask him actually. So thank you for telling me. Yeah, I grew up in Germany, so
You are
part of
German.
I'm Portuguese. I was born there by my stepfather. He's German, and we moved to Germany. So
I see.
and so my brothers are still in Germany, and my mother is, my parents are half time
I
in
see.
Portugal, half time in Germany,
I
because
mean,
to see my brothers and the kids and so on. So yeah.
I see.
Okay.
And
Oh,
we start.
please go. No, please go ahead. Just say a word.
I was just saying, I have been in Germany for only 8 years, less than 8 years. So, not I cannot speak German so far.
Oh, 8 years? Yeah,
Yeah,
it's
but
a hard language to learn. It's
yeah,
not easy,
yeah,
I think.
it's very difficult.
I was just lucky that I moved as a child. I was in second grade, so 8 years old or so. So it was intuitive learning, right? So it's a different type of learning. There was no German for foreigners class in Germany. I was just thrown into school and say, get along and cope with it. But it's a very efficient way to learn. You have no
It
choice.
definitely, yes, that's true. For me, is unfortunately do not have this chance. So
Yeah,
my
because
all
people speak
working
in,
experiment only speaking
yeah,
English. So
yeah, they all speak in English with you. So what helped
yeah.
me a lot is to, so my parents bought me these stories to listen to in
Ah,
German, and I listened to it apparently all day, and then I just started talking with the story, and apparently I learned it really fast that way. So I don't know if you can watch the TV with subtitle in German, with English
uh,
subtitle, and then maybe that helps. That's how I learned English also as a kid. Like in Portugal, there was never translated version of the movie. It was always just English with a subtitle, so you kind of learn the language that way.
oh, I see. I mean, I grew up in China, and all of our animations or movies are translated to Chinese, so don't have a
Yeah,
chance to.
exactly. Yeah, that's
Yeah,
in Germany same way, right? So that's why
yeah.
Germans
They
are
all,
not good
yeah,
English speakers generally. I mean, there
yeah,
are a lot of good but there are not really a lot of kids that just intuitively. So yeah, maybe you can try it. It worked for me really well, but I
yeah.
was a kid also, so it's different. I think now I wouldn't be able to. Okay. So, yeah, welcome everyone. The swim or sink method of language learning. Yeah, exactly, Peter. That was with me. And the funny thing is, I learned it really fast, so the teachers would still say, "Oh, she doesn't know German. You can't expect that from her." And I kind of took advantage for like the first half year.
That's good.
Okay, so welcome everyone to Science Society, and of course, a special welcome to you, Yao. And before we start, let me introduce our guest speaker to everyone, and we go from there. So, Dr. Siyao Wang, she is an independent PI at the Institute for Genome Stability in Aging and Disease at the Research Center of the University of Cologne. And she did her bachelor's degree in medicine in China, and she then did her PhD training at the University of Manchester in UK, and later on her postdoctoral research at the IGSAD at the research CECAD Research Center in Germany, and she mostly is interested in her research on epigenetics, DNA damage, and aging. And she uses C. elegans as a model organism, and she has uncovered role of histone modifications in aging in response to DNA damage, and also paternally inherited genome instability, which I think is really interesting. So really thankful for you to come here and discuss this research with us. It's really an honor. And
Thank
before
you.
we start, we conduct like we do a more interactive, introductive way.
Mhm.
So, how did you become a scientist? Like, was that something? Was it a dream of yours, or was it something that came later? maybe something interesting or a supportive teacher that guided you to this field? Thank you.
I think that is a very big question. I mean, I always told all my colleagues or PI, because they are not familiar with Chinese environment. I mean, in China, most of the kids, when you ask them what they want to be when they grow up, most of the kids will say they want to be a scientist. It becomes like I don't know, a tradition. And when I was a kid, I also gave this answer. I don't want to be different from other. But since I become a teenager, and then I gain my interest in biology because of, I think during I'm 13 or 14 years old, and I think I start my interest from the stem cell. I don't know why I just attracted to this idea about stem cell, and then DNA, and then since I start my medical training when I went to my university, and then I decided to go for more research related instead of clinic related, and I involved in a project, a very small project when I was a bachelor student, which is about microRNA and also RNA interference technique. And that is when I start looking at epigenetics. And I wrote a very small review in Chinese about this small interference RNA. It's let7 family when I was in bachelor. And that is actually the beginning of my epigenetic career, I think. And then after I finished my bachelor, I start to apply for PhD program in UK. And when I had my interview with my previous PI, he asked me, what do you want to do in your PhD? I just told him, I want to do epigenetics. I like this field. I think something on top of genetics, because I mean, we know a lot of things about genetics, but there are so many things that can regulate this genetics information on top of it, which we don't know. And that basically gave me the motivation to go to this direction. And my previous PI in Manchester, he is really impressed, and he even told me this story when I graduated. He said when I had an interview, he's really impressed by what I know what I want to do. And then after after I was doing my PhD, I gained the experience on epigenetics, especially histone modification, and I also gained my experience on C. elegans, and that is what I plan to focus in the future, epigenetics and something. But when I was doing my PhD, it's mainly developmental biology. I was also fascinated by that. So that's why I also quite interesting about the embryonic reprogramming and how this epigenetic modifications decide the cell fate during embryogenesis. That is also a very interesting question that I feel maybe in the future I will focus on that a bit more. But after I finished my PhD and I joined Björn Schumacher's lab, as you may see, I'm like a very junior PI. I just get my position last year. Before I was a postdoc in Björn Schumacher's lab. And when I joined this lab, Björn's lab is mainly focused on DNA damage. And that's why I told him that I want to bring my background in epigenetics to the DNA damage field and see what's going to happen in this combination. And that's how I developed everything.
Wow, that is so impressive that so early on as a bachelor student you also already found the path or the field that you thought was interested in, and that you then wrote this paper. That's very impressive. I agree.
Yes, at that moment, as a bachelor student, writing a review is very rare. So
Yeah.
I feel like that's actually the beginning why I interested with epigenetics is all because of that. For writing that review, I read a lot of paper, read a lot of literatures about epigenetics. I feel like it's a very cool idea. That was like more than 15 years ago. So was
Yeah,
like that is
yeah,
when I really start my interest, I think, to do this.
yeah, and that's really early on also for the field, right?
Yeah,
Not just for you from the personal perspective, but also for the field to be so early on
yeah.
interested in it and to know already as a bachelor student about the field is, I think, very impressive. So I'm so glad you did and you had the opportunity to write because now you make these new discoveries that kind of change our theories about inheritance and you know many more things you do relevant that are very relevant for aging. So I'm so glad and that you did that. And so for this specific paper,
Mhm.
how did you come to study paternal contribution basically of epigenetics, and did people believe you? Like I mean, I'm sure you were surrounded with supportive scientists around you, but when you wrote grants or when you presented this somewhere else, did people how did people respond, and was it easy to start this, or was it very very tough?
No, it's quite tough. I mean, I have done my postdoc for 7 years, as you may see in my CV. And during the 7 years, I have conducted three independent projects. But this one, this paternal transgenerational inheritance one, takes the longest one. So the reason why I spent so much time on this because first, paternal transgenerational inheritance was not a very hot topic, and no one really look at this. And there are very few knowledge about this, and also none of them are study this in. I mean, very very few people are working with this in on C. elegans. So I had a lot of difficulties when I was conducting this experiment, conducting this project. But I can tell you a very interesting story is when I joined this lab, I didn't aim to do the transgenerational inheritance project. I mean, my background is epigenetics or histomodifications, and I was interested in how this epigenetic marker are involved in DNA damage in aging process. That is actually the main project when I start my postdoc. And because I know that the germline, the germ cell, are normally quite resistant to DNA damage. They have very nice, very efficient DNA repair machinery. And I didn't really pay too much attention at the beginning of my postdoc, and I didn't really ask the question how this transgenerational inheritance works. I mean, it all starts from kind of a small accident, not really an accident, but it's not even my accident. I mean, if probably you or any of you may know that in my lab, in Schumacher's lab, about 80 years ago, our lab published a story also on Nature. It was a very nice one. He find is a senior postdoc from our lab. Her name is Maria, and she noticed when the germline got DNA damage, it can trigger the somatic to have a resistance phenotype. So the somatics will become more resistant to DNA damage. It's a very interesting phenomenon. And she also published a very nice paper. At that moment, she has conducted a lot of germline DNA damage projects. And normally, after the DNA damage, she will keep the worm for about 1 week to observe the somatic resistance. But there was once that she forgot to throw to discard those old plates, and so she left all of these plates in the incubator for more than 2 weeks. And then once she remembered there are old plates, and she before she threw them out, she looked at them under the microscope, and then she noted one thing is when they gave irradiation, the irradiated plate shows a lot of dead embryo or dead eggs, but in the control plates, there are mainly starving worms or starving larvae. So she thought this high embryonic lethality is due to a transgenerational effect of germline DNA damage. And at that moment, I just joined this lab, and because I was the only one that have the epigenetic background, so she presented this finding on the lab meeting, and then she think that maybe my epigenetic experience or my epigenetic background can help to study to understand this phenotype, and that is where I start when I think about, oh, there could be a potential project, but at that moment I still focus mainly on my other project, which is how the epigenetic regulate aging in response to DNA damage. But since then I started working on this transgenerational inheritance, but was very difficult at the beginning because if you know C. elegans, you know that C. elegans are mainly hermaphrodites. They have very few percentage that are male. Most of the population are hermaphrodites, indicate they are bisexual. So they have both female and male reproductive system in single worm. And once you look at an adult worm, you will see that the germline, the very large germline, they only do oogenesis. They only produce oocyte because spermatogenesis have already completed before they enter the adulthood. So most of people study the germline DNA damage response are mainly focused on oogenesis. And also oocyte DNA damage also link to like a lot of transgenerational effect, also the chromosome abnormality in the progeny. So at the beginning, also Maria and I, we both think that the effect is coming from the oocyte. We never think about it's coming from the sperm. So that's why in the first year of my, when I conducting the project, I was focusing on epigenetic marker and oocyte. And that's why I make like 1 year like totally wasted, and I didn't get any positive result. And I have tested so many epigenetic marker in the oocyte and also look at the conformation change of the oocyte nuclei, but nothing was found. And I find out was paternal radiation effect was on almost the second year when I start the project. And then that is when I eventually start this project. And after I know it's a paternal effect, it's also having a lot of trouble because there is not much information about how this sperm carrying epigenetic information to the further generation. There are a few papers, but I have also tested those epigenetic marker, but none of them shows a positive effect. So was really difficult, and I was kind of almost give up in between on this project because the mechanism is very difficult to discover. I mean, the phenotype is very good, but the mechanism is really hard to find. But we are lucky. I mean, at almost the 2 years ago, we did this CELAC assay, and after we doing this omics analysis, and then find the linker histone are the key. And then we're starting to solve this problem.
Well,
but it was really a terrible, not terrible, I mean, very difficult time.
I'm so glad you got through it and you stayed persistent, and I'm also very glad that you stayed open to what the data shows you. And I think that's very important that we don't let our worldview and bias view overshadow the data, and then being persistent. So, I mean, I hope you feel now that it was totally worth it. I'm so glad. I'm thankful for you that you stayed persistent, because I think this changes our knowledge system a lot. So it would have been very, very, very sad if you would have
if
given
I
up.
give up.
Yeah. So thank you in the name of all of us, and yeah, now everyone should be able to access the paper. So the stage is yours, unless Peter, did you want to ask something before we start going into the paper?
No, I have a question or a thought that is connected to how this works with DNA and then epigenetics, which is kind of, and then how the programming of the brain works and so forth, because I think
Oh,
they are interestingly connected, and I think that one needs to look at them, split those up separately in order to have good models for understanding. Anyway, I hope that
okay.
if that doesn't make sense whatsoever, I don't have any questions or comments. But
Oh no, then just go ahead. I mean, every question is has a lot of value. So
yeah, no, I think that I want to listen to the paper first. So but that is the direction my brain went.
okay,
So anyway, thank you very much.
yeah, no problem. So maybe now I can start. So before I start the story, I can give you a very short introduction about this paternal transgenerational effect. I mean, when I start to notice it is a paternal effect, I start to read through some literatures about what the previous knowledge about this paternal effect. And then I found a very interesting studies, like in the last century. I don't know whether you know there is a nuclear power plant in Sellafield in UK, and then there are a small epidemiological group noticed that the kids that living in the village next to the nuclear power plant have high incidence of leukemia and lymphoma. And then the government sent a small research team or investigators to the village and to study what's wrong with this. And then there soon, after 2 years, there is a report coming out and leading to a theory or a hypothesis is called the Gardner hypothesis. And this hypothesis said if the father exposed to radiation within 3 to 6 months before conception, then it will lead to high risk of cancer in their children. And once they expose this report, and soon is leading to a highly controversial response because first, the N number is quite low, and also studies coming from other nuclear power plants do not show very supportive um, observation. And also, people, especially genetic scientists, trying to investigate whether the parental exposure to radiation can lead to the children gains more DNA mutation. And then they analyze the survivors from Chernobyl accident or from the Second World War, the atomic bomb. And then they find there is no incidence of the hypermutation or there is any de novo mutation change in their progeny. And then after so many years, the Gardner series got rejected, and then the official conclusion is there is no indication of transgenerational effect of parental exposure to radiation. That's what happened in humans studies. But in animal, the study shows like a very opposite and a very clear observation that in mice, that radiation in the parental generation can leading to high incidence of cancer and infertilities and also mutations in their progeny. So animal study shows very different from human study. And then it's leading to how to say, a very making this question is very controversial, and but like one of the reviewer mentioned one sentence I think is very interesting. Is the absence of evidence of an effect doesn't constitute evidence of absence of this effect. So you cannot use all of this to say there is no incidence, no evidence to support there is transgenerational effect. So that's why when I start my project, based on this question, I ask whether we can use C. elegans to answer this question. I mean, But you know that C. elegans are mainly hermaphrodized. In order to distinguish whether the transgenerational effect is coming from the father or is from the mother, we have to use sex-separated mutant of C. elegans. So in our case, we used the 2 mutant, which is FOC2 and SPY8. They have a different way to separate the sex. So the FOC2 is a one that I mainly used in my whole paper. So the FOC2 mutant has a spermatogenesis defect. So in the hermaphrodite worm, FOC2 worm, they cannot produce the sperm. So their germline can only generate oocyte. And in the male version of the FOC2, they are like the normal male. They continue produce the fresh sperm. So after we get this sex-separated system, we think that it's a perfect model to mimic the dioecious system like in human. And then we're trying to ask the next question is which gender or it's whether it's oocyte or sperm can making a transgenerational effect. So now we can move on to the figure 1. I think when you read this paper, you may find this a little bit annoying because I changed the name of all of the generations and also the treatment. So at the beginning, we're trying to ask whether irradiated mother will leading to a transgenerational effect. We call this irradiated mother as delta F, as you can see in the figure 1. And then after irradiation, we immediately made the delta F with unirradiated father. With unirradiated father. And then after mating, we immediately analyze the progeny viability. And then we find there is a dosage-dependent increase of progeny lethality, as you can see in the figure 1a. And this increase is due to that there are so much damage happened in the oocyte that cannot be repaired. But this damage will lead into DNA fragmentation, and that will be passed down to the embryo. And the embryo, when they are doing the embryonic embryogenesis, this damage or this DNA breaks will leading to mitotic catastrophe, and that is why it's leading to this dosage-dependent increase of progeny lethality. So that's why in the F1 generation, most of the progeny are dead at embryo stage, but still you can find some of the progeny are surviving, especially in the low and the medium dose group. And when we maintain this survived progeny to adulthood, and we call the daughter of irradiated mother as delta FF, and the son of irradiated mother of delta FM, and then we mate them again with unirradiated countersex, and then we measure their progeny lethality. And then we find that this survived the delta MF and delta MM, delta FF and delta FM, their progeny are totally fine. So there is no transgenerational lethality from irradiated mother. And then we move on to the next question is, how's the father? And then we only irradiate the father. We call the irradiated father as delta M. And after irradiation, we made them again with unirradiated countersex and measure this progeny viability. And surprisingly, unlike the irradiated mother, when we only irradiate the father, the progeny lethality is relatively quite low and do not show a dosage-dependent manner. So they are roughly around 10 to 20% of progeny lethality. Most of the progeny are healthy and viable. So we maintain the F1 generation's progeny to adulthood, and we call the daughter of delta M as delta MF, and the son of delta M as delta MM. I know that the names are quite weird, but please keep this name in mind because I will refer to them throughout my talk. So after I make them with also unirradiated countersex, I measure their progeny viability, and then the surprising thing happened. It's the progeny viability of delta MF, the daughter of irradiated father, reaching to almost 100% lethality. Almost none of their progeny can survive. and the progeny of delta MM, the son of the irradiated father, reaching to around 50 to 70 of progeny lethality. That's very high. So this high progeny lethality that we observed in the delta M in the F1 generation indicate irradiated father can lead to transgenerational lethality. And that is the first phenotype that we observed in this project. And the next question that we ask is how many generations this effect may last? And then to answer this question, we pick the very rare survived F2 generation that even in the delta MF progeny, there still have like very, very few progeny that can survive and become adulthood. We also gave them name and made them with unirradiated countersex. And then in the F3 generation, when the F2 gave progeny, and then the F3 generation or the progeny of F2 shows normal viability. So indicate that the paternal transgenerational effect can only reach to 2 generations. So the progeny of the F1 or the F2 generation will be the main generation that take this effect, but not even longer. So after we confirm that the paternal radiation will leading to a transgenerational effect, then the next question is which stage of the sperm are responsible for this effect? And now we move on to the figure 1C. And as you may know that like in human, the sperm are continue produce refresh themselves. So they keep producing new sperm. I think in human, the whole process may taking about 65 days. In C. elegans, is much shorter. And in C. elegans, is also they keep producing new fresh sperm. So in order to understand which stage of the sperm can carry this transgenerational effect, we basically just made the irradiated male at a different time with unirradiated female. So immediately after irradiation, we made them with unirradiated female, and then we perfectly capture this transgenerational lethality. But if we wait one more day and transfer the same irradiated male with another group of unirradiated female, and then this female will using the sperm that was immature when they are getting irradiation. And then we find there is no transgenerational lethality. And this experiment told us only the mature spermatides are responsible for carrying this transgenerational effect. But immature sperm or spermatogonia or spermatocyte, they can still repair the damage. So this is basically what figure 1 is, but just in parallel, we have done all of the experiment in hermaphrodites, which is a wild-type C. elegans as well. And unlike this sex-separated mutants or this dioecious system, hermaphrodite C. elegans do not need to mate with a countersex, so they can just reproduce themselves. So there is no outcrossing. So in hermaphrodites, this transgenerational effect can last much more generations than in this FOLK2 or SPE8 mutant, because that is what we found at the end is because the effect of this transgenerational lethality can be diluted by crossing with unirradiated countersex. If you have never mated with unirradiated countersex, then you cannot introduce intact chromosome to this animal, and then this effect may last much longer compared to the FOLK2 and the SPI8 mutant. So after, in the figure 1, we know there is a transgenerational effect from the father irradiation exposure. The next question that we ask is what kind of DNA damage that inherited from this radiated sperm. So to understand this, we decide to use staining to directly tracking the chromatin conformation immediately after irradiation. So if you move on to the figure 2, you can see that immediately after irradiation, the sperm shows a morphological change. So instead of this round shape, it becomes more like a comet shape. And that shape indicate those sperms are carrying DNA double-strand break. So their DNA are basically fragmented because we use quite high dose, as you may see. And however, this is not really a surprising thing, but those sperm carrying DNA double-strand break do not affect their fertility rate. I think it's already known in mammalian system or in other animal, sperm carrying DNA damage, especially mature sperm, do not affect their fertility. So they can perfectly fertilize fertilize an egg. And after fertilization, in our case, most of the embryos are viable and they can still hatch and develop to become a healthy adult. But in the embryo, we also observed a lot of characteristics about genome instability, such as the chromosome lagging and the chromosome bridging. And the chromosome bridging phenotype we can also see in the somatic tissue of the F1 adult. So the irradiated father's daughter and the son, they also are having this chromosome bridging phenotype in their intestinal tissue. So why the chromosome bridging is very interesting. I think maybe you may know that the chromosome bridging is normally indicate they are breakage fusion bridge circle. And that is a circle that normally happened in cancer cell. It's normally a signature of the cancer cell that their genome are super unstable. So they are keep making new double-strand break, and those double-strand break will be mistakenly fusioned together with a wrong break end, and that normally will leading to very carcinogenesis phenotype. And we found this in the F1 generation, no matter in the embryo or in the adulthood. And that indicate those F1 generation, the daughter and the son of the irradiated father, are keep generating new DNA breaks or DNA fragmentation or new DNA chromosome fusion. And then when these things happen to the gametogenesis, when they're starting to generate germ cells, go through meiosis, the bad thing will happen because they cannot pairing, because the paternal DNA are broken. They are carrying double-strand break or misfusion, so they cannot pair with maternal intact chromosome. They cannot find their pairing, so they have a pairing defect. And also by doing staining, we found those germ cells that during crossover, it's called Parkinson stage, we found a lot of DNA fragments by staining. At the beginning, we're still thinking these fragments are due to the misfusion. So we're trying to use a telomere fish. We're directly trying to monitor the telomere at the two ends of the chromosome, but we found that a lot of these DNA fragments do not have telomere sequence. So they are like completely just break the DNA. So after we found this DNA breakage in the germ cell, and then we also notice they cannot pairing properly, and that's perfectly makes sense why their progeny are dead, because their germ cells are mainly aneuploidy. So instead of having 6 pair of the chromosome, they have irregular number of the chromosome in the germ cell. And in C. elegans, aneuploidy leading to very high level, almost 100% of progeny lethality. And that's why it's explained why we can see the F2 generations progeny lethality. In order to directly visualize this chromosome fusion or chromosome bridging, we decided to perform a single worm whole genome sequencing, so we can directly see their sequence, how they are fused together. And to do the single worm whole genome sequencing, we pick 10 from the delta MF group, which is a daughter of the irradiated father, and the 10 delta MM, the son of the irradiated father. And in the figure 2G, here is an individual example from each group. And this is our sequencing result. as you can see in this form, they form a lot of this chromosome misfusion, or we call this in our paper as chromosome translocations. So what are they? So you can see that on the graph, part of the chromosome 1 translocated to chromosome 5, part of the chromosome X translocated to chromosome 3. So this is what we call the chromosome translocations. And we find another interesting thing about when we are looking through our translocation map. We found another interesting thing is if you look at our supplementary figure, you may also notice all of the delta MF, all of the daughter from the irradiated father are carrying at least one translocation on their X chromosome, but all of the worm that belong to the delta MM group, that the son of the irradiated father, none of them carrying translocation on their X chromosome. I mean, that could be very easily explained because their son, the irradiated father's son, the delta MM, can only inherit an X chromosome from their unirradiated mother, but their daughter will definitely inherit one X chromosome from their irradiated father. So that's why all of their daughter carrying translocation on the X chromosome, but none of their son carrying translocation on the X chromosome. And then it reminds us the phenotype that we observed in the figure 1, that the daughter of irradiated father shows much higher, much severe progeny lethality compared with the son of the irradiated father. So we were wondering whether this difference of their progeny viability is due to the X chromosome translocation. At that moment was really very lucky. At that moment, there is one paper that just come out in Elegans. He found, this paper's author found like X chromosome integrity can affect a sex chromosome transcription inactivation, which is happened during meiosis. This process is very important for the during meiosis. If the X chromosome are desilencing, so they cannot be transcriptionally silenced, then it will leading to very high level of progeny lethality. So we are wondering whether in our case, the translocation happened on the X chromosome can leading to high progeny lethality is due to the transcription activation in this X chromosome. To prove this hypothesis, we perform a staining with antibody against transcription activation marker. It's phospho-SIR2, the SIR2 phosphorylation on RNA polymerase 2. And we also co-stain with X chromosome marker, which is a Hem8. So you can see that in the panel H, that the chromosome that labeled with the red dot, which is the X chromosome, the red dot is Hem8, is the parent center protein of the X chromosome. And this chromosome are normally silenced because of the meiotic sex chromosome inactivation process. But in the daughter of the irradiated father, this chromosome shows transcriptionally activation, and the transcription activation on the sex chromosome are known that in their paper linked with high progeny lethality. So that perfectly explained why their daughter have much severe transgenerational lethality compared with the son, because the daughter carrying X chromosome translocation. So after we understand what kind of DNA damage are transmitted from the irradiated father, and the next question we ask is why the double-strand break happened in the paternal genome cannot be properly repaired in their progeny or during embryogenesis. So we want to know what kind of DNA repair they use, because this is definitely a wrong DNA repair machinery because they make so much mistakes. And when we are analyzing our DNA sequencing data, we're specifically looking at the translocation site. And because when we're doing single worm whole genome sequencing, we cannot see DNA fragmentation. We can only see this translocation. So we look at the break points of this translocation. And surprisingly, at this break point, we found that there are templated insertion. This is move on to the figure 3. And this templated insertion, what is that? Is about a short base pair that has a template within a very close region to this breakpoint. And this template are inserted into this breakpoint. And this is called templated insertion and insert. And these inserts are normally is a typical signature of a special DNA repair machinery. It's called alternative non-homolog end joining or polymerase theta-mediated end joining. Another feature of this DNA repair is they are normally using microhomology as a template to annealing the double-strand break. So here I can give you a short introduction about how to repair double-strand break normally. So there are two classic DNA repair machinery to repair the double-strand break. Normally, one is called homolog recombination. It's an accurate repair machinery because it's always using the homolog chromosome as a template, and then they repair this double-strand break based on the template. So that's why it's an accurate, it's a precise, error-free DNA repair machinery. And the other classic repair machinery is called a non-homolog end joining, or NHEJ. And this repair machinery do not use a template. So they directly annealing two break end together. So when they are using NHEJ, they normally making mistake. But the TMEJ, the polymerase theta-mediated end joining, which I found in our paper, is an alternative end joining. It's not belong to homolog recombination. It's also not belong to the NHEJ. It's an alternative way. It's normally activated when homolog recombination and non-homolog end joining are both inactivated, and then the cell may use the TMEJ, the post-theta-mediated end joining to repair. So this repair machinery, instead of using the homolog chromosome sequence as a template, they're only using one or two base pair of the homolog as a template. For example, the break end, they use only the one base pair as a homolog, as a template. So they're trying to use template, but they use very short template. That's why it's an error-prone repair machinery. It's also making a lot of mistake. And what we found in our case, in this F1 generation, we found this F1 generation also have this microhomology pattern, as you can see in the panel B. And that indicate instead of using homolog recombination, the HR, or using NHEJ, they are actually using this TMEJ to repair this paternal DNA damage. And that also perfectly explains why they are keep making new breaks, because none of these repair are accurate. They are keep making new misfusion. And that is move on to the panel C. So the panel C is, once we found this TMEJ marker, we want to know whether TMEJ is the only repair machinery that was engaged in repairing this paternal DNA damage. We're screening through different mutants from the homolog recombination, non-homolog end joining, and the TMEJ, and we want to find which mutants may leading to like more sensitive to this paternal DNA damage. And the result is very, very obvious. Like when you can see that the BRC1 cross with FOLK2, we all cross them into the FOLK2 background. The BRC1 is a mutant of homolog recombination, and the CKU70 is a mutant of non-homolog end joining, the NHEJ, and the POQ1, the POLQ1 is a mutant of the TMEJ. And we use these three pathways mutant, and we found when we mutate the homolog recombination, there is no significant difference. when we mutate the non-homolog and joining the CKU70, there is a slightly increase of protein lethality, but it's mainly at a high dosage group and also in the progeny of delta MM. But surprisingly, when we remove the theta-mediated repair protein, which is the POQ1, then the F1 generation already shows almost 100% of progeny lethality. And also the survived progeny are sterile, so there is no further generation. So that clearly shows that only the paternal DNA damage are mainly using this alternative NHEJ or the TMEJ to repair this paternal DNA damage, which is the post-theta-mediated and joining. And because this is an error-prone repair machinery, that's why it's leading to all this genome instability phenotype that we showed in the figure 2. And the next question that we are curious is whether it's the father, the sperm itself, can repair their damage within the sperm by using TMEJ, or it's a mother that after fertilization can repair the paternal DNA damage with the TMEJ. So to answer this question, we use different combination of the mutants that either on the father or in the mother, and then we find, like, as you can see in the FOG2 single mutant, their F1 generation can only lead to around 10% of progeny lethality. But when the father are devoid of this TMEJ protein, POQ1, then it's also leading to almost similar level of as the FOG2 single mutants. But when we remove the POQ1 in the maternal, in the oocyte, and then it's leading to almost 100% of progeny lethality. So what does it mean? It means that the paternal DNA damage cannot repair their own damage within the sperm. It can only be repaired after fertilization and the use of paternally provided DNA repair machinery, which is a POQ1, the TMEJ, to repair the paternal DNA damage. So now we know that paternal DNA damage can be repaired by using an error-prone repair machinery, which is TMEJ. And then it's raising the next question, which I think is also the most important question. Why the progeny decide to use an error-prone repair machinery to deal with the father's DNA damage? Why they cannot activate accurate repair machinery? So to understand the mechanism, we actually struggling with this question for several years, but we performed an omics study at the end by using the CELAC. And so with the CELAC, we trying to see in the F1 generation how the proteome change and which proteins are increased, which protein are decreased in the F1 generation. And then we found, like, in the F1 generation, as you can see in the figure 4 in the panel A, we found most of the protein that changed are within this protein DNA complex assembling process. And if we look at all of this protein involved in this process, we found in the panel B, most of these protein are upregulated in the F1 generation. And the top 3 candidates are the histone H1 proteins. So what is this histone H1 is doing? And at that moment, we are not sure what are this increased histone H1. So we decide to knock them down. So we're using the RNAi to knock down this histone H1 protein, the linker histone proteins. And then we found, like, at the beginning, we knocked them down in the hemophytides, and then we found that there is a residue effect on the transgenerational lethality. And then we generate a mutant, also crossed with a FOG2 mutant, and then we found the mutant also significantly rescued the transgenerational lethality, which indicate that the increased linker histone H1 is a reason that leading to this transgenerational lethality. So why is histone H1 is so important? Then we should going back to the function of the histone H1, what they are doing. So normally we know that increase of the histone H1, because it's a linker histone, is normally linked with a conformational change of the chromatin. It can make the chromatin more compacted. So another important thing is when you increase his heterochromatin, you increase the histone H1, it's also leading to the heterochromatinization. So the chromatin becomes more heterochromatin-like. And in order to confirm whether increase of histone H1 is correlated with increased heterochromatin formation, we also performed the heterochromatin marker immunofluorescence. And as you can see in the panel D, in the daughter and the son of the irradiated father, the delta MF and delta MM's germline, they shows much higher level of the heterochromatin marker, which is H3K9 ME2. That is the marker in elegans for heterochromatin in germline, and we can see there is significant increase. And the increase of this heterochromatin is not restricted to specific chromosome. It's basically quite random on the whole genome. And then we want to know whether by removing the histone H1, we can diminish the heterochromatin level. And then in the panel E, we perform the histone H1 knockdown, which is a His24 knockdown. And we can see that there is significantly reduction of the heterochromatin level. And in parallel, we also knock down the heterochromatin protein, which is HPL1 in C. elegans. And we also find that there is a significant reduction of heterochromatin formation. As you can see the quantification in the panel E. And then we are wondering whether directly removing the heterochromatin formation by removing the heterochromatin protein can also rescue this transgenerational lethality. And then the answer is yes. We found that in the panel F, we also performed this transgenerational lethality assay, and in this HPL1 cross with FOC2 double mutant, and then we've also found a significant rescue effect. That indicate the increase of linker histone and heterochromatin formation. It looks like it's a barrier for survival of the F2 generation. And then we are wondering why making the chromatin more compact can make the progeny lethality. What's the reason? So when we are performing the experiments in the figure 4, when we are doing the staining, we also noticed the one thing is the homolog recombination marker, which is the chromatin is a RAD51 that we used as a homolog recombination marker. Homolog recombination marker is increased in this His24 and HPL1 RNAi group. So what does it mean? So homolog recombination is accurate repair machinery, as I introduced to you before. And this marker do not show a severe increase in the F1 generation in the normal, in the empty vector treated group. However, in the His24 and the HPL1 RNAi group, when we remove the heterochromatin and remove the linker histone H1, we can see in the germline, this shows increased level of this RAD51 foci, which indicate those germ cells can activate an accurate repair machinery. And then we want to know whether the activation of the accurate repair machinery is the reason they rescued the progeny lethality. So therefore, we performed our, actually the last experiments, we combine all of this knockdown. So we knock down both heterochromatin marker, histone H1, and also the homolog recombination protein BRC1. And then, as you can see, when we only remove the BRC1, the homolog recombination protein, they do not show any change compared with the empty vector, which is previously known because paternal DNA damage do not use homolog recombination. And when we remove histone H1, or when we remove heterochromatin marker, this shows a rescue effect. But interestingly, when we remove both the heterochromatin and the homolog recombination protein, then the rescue effect is completely abolished, which indicates the rescue effect of removing the histone H1 is based on the activation of the accurate repair machinery, homolog recombination repair. So basically, that's the end of my talk today. And the last figure is a summary of the model of our story. So it's very simple. So after irradiation on the male, the sperm DNA becomes fragmented. However, it doesn't affect their ability to fertilize the egg. And then after fertilization, the oocyte or the maternal side, we're using an arrow-prone repair machinery. It's called theta-mediated, especially the polymerase theta, come to repair the paternal DNA double-strand break. And because this TMEJ is arrow-prone repair machinery, so they keep making the DNA, the chromosome misfusion or chromosome translocation and DNA fragmentations in those embryos. Once this embryo grow up and start to generate germ cells, and that things will also making mistake during meiosis, and also importantly in the germ cell, this misfusion or chromosome translocation or DNA breaks, making this chromatin becomes more compacted. So they recruit a lot of histone H1, building this heterochromatin formation, and then this heterochromatin formation block the usage of the accurate repair machinery, which is homolog recombination. And because it's blocked the accurate repair machinery, so their germ cells are mainly aneuploidy, and this aneuploidy leading to this high embryonic lethality in the F2 generation. And here has a two scenario. One is a daughter, one is a son. And the son, because the X chromosome are still perfectly silenced because the X chromosome are intact, so their progeny lethality is milder compared with the daughter. So basically, that's what I found. So now, please, if you have any question, it's time.
Thank you so much. This is such beautiful work, and thank you so much for presenting it to us. Because
Thank you.
I can only imagine how much work it was. It must have been a lot of work. And the mechanism you found is really interesting. Just, when you started, also how you showed again more like a principle, like how for evolution it was really important to have female and male, because you showed that when you separate C. elegans, male and female, this just goes for 2 generations. But then if you don't have that, this damage goes on and on and on. So
Yeah.
already that is a really, that you show that in a model like that is already really important.
I think the important thing, I mean, if you are mentioning in the C. elegans, normally they are hermaphrodites. So the progeny of the carrying that paternal DNA damage, they can carry on this DNA damage forever, because after the F2 generation, the DNA damage that in the progeny becomes stable. They are aneuploidy. Aneuploidy, although they are leading to high lethality, but they are more stable compared with in the F1 generation. They are basically making new breaks, so the genomes are unstable. So that's why in the F2 generation, most of them are kind of eliminated. The one that carrying very severe chromosome abnormality are eliminated, and that's why the progeny can still survive. Survive the one, they can still carry normal progeny that carry on. They can still survive, but in the hermaphrodites, when they cannot, mated with another pair of intact chromosome, then this phenomenon will continue forever. So in the hermaphrodites, I think it's kind of benefits of the evolution. It's this it's the dioecious system that they are keep mating with unirradiated or undamaged countersex, can making the species to eliminate the detrimental effect of the genetic mutation or genetic chromosome abnormality, and then they can maintain the species. But in hermaphrodites, in C. elegans, like this bisexual animal, without this mating system, a lot of this damage will be continue forever, like much longer.
Yeah, exactly. Already that I think that you show this so this evidence in such a great way. Already that I think is really interesting. And then on top you show the mechanism, the repair mechanism, and which parts are essential. That is also really interesting. And
Thank you.
yeah, so but before I go ahead and ask a lot of questions, I wanted to ask Peter and LT if you have a question that you wanted to ask.
I think I wait a little bit and think if I have a better question. Yeah.
Sure. LT, did you have a question?
Yes. Have what histone 1, right, knockout? Is there any other histone things you tried to knock out to see whether the
Yes.
effect would be similar? Thank you.
Yes. Actually, after we got this proteomics result, we noticed there are a lot of histone proteins are upregulated in the F1 generation. It's not only the histone H1. Actually, the histone H1 is just the top candidate. We also trying to knock down all the other histone proteins like histone H3, H2A, H2B, but actually none of them can show in this rescue effect. Histone H1 gave me the best result. And it's not only the His24, as you can see in our paper, we mainly use His24 as H1 knockdown. But we have also tried of HEO3 and HEO2, which is also another two protein encoding the histone H1 protein in C. elegans. And HEO3 also shows a partial rescue effect, but the effect is much weaker compared with the His24. And HEO2 is very lethal. When we knock down HEO2, the worm cannot generate a viable progeny. So I think also happened when we knock out other histone proteins, some of them leading to very high level of lethality. So I cannot get a clear conclusion from that. But His24 is a very clear one.
Because I was almost thinking, because you have done some double knockout, I was just thinking, so now I understand.
Yeah, we
So
also
maybe
did that.
others too lethal. Yes, thank you. Yeah.
Yes, we also have done that. HEO2 is too lethal. HEO3 and His24 double knockout, making a better rescue. That's true.
It's beautiful. I have to go back and read more because
yeah.
I missed the beginning. Your introduction is important. Thank you so much. Okay, back to you,
No problem.
Katerina. Yeah.
Yeah, thank you so much for those questions. And I wanted to check with you the time, how much time you still have for questions.
Oh.
Do you have like 10 more minutes, 50, just
Yeah,
to
sure.
Yeah.
Sure.
Okay,
yeah.
perfect. Yeah, thank you. And I wanted to ask if do you also check for other epigenetic mechanisms, or are you planning to check such as methylation effects, or also maybe 3D formations that maybe also happen to maybe milder radiation? I don't know if if strong radiation would change any of that, but maybe milder ones. Thank you.
Yes, Oh, that's a good question. Actually, at the beginning, we are mainly focused on epigenetic modification, so that's why we do tested quite a lot of epigenetic modifications, and especially as you may know that in C. elegans, one of the modification is quite correlated with transgenerational inheritance is H3K4 methylation. We have focused on this marker for quite long, but so far we didn't find any evidence about this modification is involved. And the other thing that you may notice that our mechanism, this transgenerational effect, is not a pure epigenetic effect. It's like half genetic or half and half epigenetics. I would say in that way. I mean, the genetic effect is the main trigger of this transgenerational thing because they are using a different DNA repair machinery. It's a very problematic DNA repair machinery. That's the main reason that it triggers this transgenerational effect. But I was also thinking whether this choice is due to some epigenetic reason, but I haven't found any evidence. But one hypothesis or just speculation is, as you may know, that chromatin of the sperm are highly compacted, highly packed. So they are replacing their classic histone with a protein called protamine. So they are not packaged with the histone; they are packaged with the protamine. So when they fertilize a sperm that fertilizes an egg, the protamine need to be replaced with heterochromatin, with a histone. And this replacement of the protamine with a histone will be one of the triggers that making the sperm DNA damage cannot be properly repaired as the maternal DNA damage. But this replacement or the 3D structure that changed during fertilization will be one of the interesting topic to follow up. I would say that.
Yeah, thank you. That's really interesting. And did you or are you planning to maybe use just human cell, like if you compare this repair mechanism and if you irradiate human cell lines, uh, is there a comparable repair mechanism that is being used by human cells? And would you expect that maybe human cells have a better strategy to repair, and that's why in the human studies we didn't see that huge effect on transgenerational damage?
Unfortunately, that so far our evidence shows the opposite. It seems like the human doing the same as the elegans. I mean, we do analyze the human data set that are in our supplementary figure 7. If you can see that, in the human data set, it looks like the paternal de novo mutations also carrying a very clear pattern of microhomology that indicate that in human, paternal DNA damage also will using TMEJ, this problematic repair machinery, to repair their damage. So it looks like it is a very conserved mechanism. And the other thing that we want to say is why in human epidemiological study do not generate a supportive conclusion is because our result finding that only the mature spermatids can carry this transgenerational effect. In human, mature sperm are stored in human in a very, very short time. What indicate is like they must get have the conception within very short time after irradiation, then it may lead to this transgenerational effect. Spermatogonia or spermatocyte, they can repair the DNA damage properly, and that is already been known. That is already known that by other people's work that the spermatogonia, they have the very good DNA repair efficiency, but only when they become spermatide, and then they lose all the repair machinery. And I think that is the period that they are very vulnerable to DNA damage and can trigger this transgenerational lethality or transgenerational effect. So I think in human, it's basically, so far, our evidence thinks is like very similar to C. elegans, that it may also trigger a similar effect. But I think if you're thinking from evolutionary view, then it makes sense. I mean, normally the sperm that brings more de novo mutation to our offspring. So the diversity of our species is actually coming from the father rather than the mother. Because the sperm DNA damage, when they have a break, no matter coming from replication, coming from DNA damage agents, this damage cannot be perfectly repaired in sperm. And this damage in human or in other species can trigger this diversity of the genetics, genetic diversity in every species, and making this population is more, they can be viable in different circumstances. I think in evolutionary view, it all makes sense.
That is really interesting. And you know, there has been some evidence or some studies showing that if older fathers have children, that the occurrence of autism, even with younger women, that the occurrence of autism is higher in the children. So do you think that you know, that this principle is not just you know, with radiation? Do you think that damage due to aging in general would the repair mechanism, would it be still the same, and
Yeah.
would it miss then a lot of damage from the aging male?
Yes, I mean, the aging is definitely a very big factor for affecting this DNA mutation rate. I mean, the older the father, they accumulate more DNA damage. That is already been found, and as you mentioned, that is correlated with mental diseases such as autism. Yeah. And we also think that is correlated with decline of the DNA repair efficiency in the spermatogenesis. And that is actually already known that in human, that along with age, the DNA repair, and also because sperm went through so many rounds of proliferation, and the DNA replication is a big source of DNA mutations. They keep making mistakes, and the repair machinery or the proofreading function of the replication also start to make more mistakes when they get old. And that is a big source of mutation come from the father. And the other thing is also chemotherapy or radiotherapy or some like occupational exposed to the DNA damage agents is another source of getting more mutation in the paternal genome. And that is also, yeah, very, very dangerous if you're thinking about your progeny's health. Yeah.
Yeah, interesting. Thank you so much. And somebody joined the stage. I'm not a legaler. Yeah, please
I
go ahead
will.
with your question.
Hi, my name is actually Soren. I haven't had the chance to change my name back on Clubhouse. To me, I'm a psychologist and a medical student, and I wanted to ask. First of all, I'm grateful for your club and for your science foundation. I was looking in the last year for scientific rooms and groups. I'm the leading expert for science, psychology, neuropsychology in Germany, in Clubhouse, not in the entire country. So I'm very thankful for all the conversations and discussions you're hosting, and it's a very fruitful and very good room. Thank you for that. Then to my question, with the since histones, you know, in the DNA, in genetics, there are different kinds of mutations, and I wanted to ask if it's known what kind of mutation is responsible for the repair restriction. Is it a silent mutation? Is it the frameshift mutation? Or because there are like different variants of mutations, can anyone relate or help respond to that?
May I repeat your question? You asking what kind of mutation can leading to the DNA repair deficiency? Is this what your question?
Yes.
I mean, in C. elegans or in human, that's a different question, I think. In human, there are quite a lot of mutation happened with DNA repair machinery, and a lot of them are linked with cancer. I think if you can see that no matter on P53 or BRCA1, those mutations are affecting this DNA damage response. But the mutations that correlated with my topic today, with like paternal DNA damage, especially POQ, the TMEJ repair machinery mutation, to be honest, I haven't found the one that in human. So I couldn't give a clear answer on this.
Okay, so this paper is not related to humans. I'm sorry, I just overflew it.
Yeah, I mean, in human, I haven't found the POQ1 mutation that related with disease. I think it's basically because POQ, this is the enzyme that is required for repair this paternal DNA damage. It's also the enzyme that required when the other two repair machinery are inactivate, and then this repair may play a role. So I think the function of this repair machinery in a normal human, like in physiological way, is not like very detrimental. Even the human carrying a mutation doesn't relate it to obvious disease. I think that's the reason why we haven't discovered like a mutation on this protein that correlated with disease. But yeah, that would be a very interesting question.
So yeah, so from what you just that answers to the question, that may assume that because in humans, so we have the early two, like the precise repairs, and then what's the second one? It's already enough. So that's why it's like underneath. It's like never has a need to activate it. So the mutation spectrum would be wider than if you look. Can you, if you have time, I'm looking at the slide 7 of your supplemental. Before, if Peter has question, Peter can ask. But if Peter is not, do you mind? Like, because
Go
okay. Could you go quickly over? Thank you.
You mean the supplementary figure 7?
Yes, because you mentioned that when Katrina is asking you a question about have you looked at the human, So either way, choose however you want to answer. You answer about the human never have the need to activate this
I see.
TMGC.
I mean,
thank you.
TMEJ. Yeah, this is the repair machinery is called the TMEJ. First, the TMEJ was like just been found like less than 10 years. It's a very new repair machinery, and for people that who discovered this machinery is when they removed like both homolog recombination and non-homolog unjoining. Then they found okay, now the cell can using another it's also called alternative repair machinery. It is TMEJ to repair the double strand break. And then they are thinking maybe this one is not the crucial repair machinery that in the tissue or in most of the tissue. But after they discovered the TMEJ, they found this TMEJ also play a lot of role in immune cell. I didn't focus on the immune cell, but I read some article about this TMEJ play a role in T cell. So I think it's also crucial for very specific event in the organism. For example, immune system, or in our case, is paternal DNA damage, but doesn't mean that when the human carrying the POQ1 mutants are completely fine. I don't think they are completely fine, but it's probably just not that obvious as when they mutate with homolog recombination or non-homolog and joining the two classic repair machinery. Maybe they can making that the progeny have a higher mutation rate, but we haven't tested this. We don't know whether in human, if they are carrying this POQ1, they are having higher mutation rate. We don't know. We do not have the, how to say, the sample to
Data
investigate
set, the
this
data
question.
set, like here we're talking about the
Yeah.
data. Yeah, thank you.
Yeah, thank you so much for those questions. Does anyone maybe? Oh, yeah. Hi. Yuan. Did you want to ask the last question? Thank you.
Oh, yes, thank you very much for giving me this opportunity. Also, I would like to thank Dr. Wang for this wonderful presentation. Actually,
Thank you.
I'm not, my major is electrical engineering. I'm quite a layman in this field, so forgive me if I ask something very fundamental.
No problem.
So my question is, you mentioned three mechanisms for repairing the DNA. And I would like to know when do they, in which scenarios do they take action, or they all start all together?
No.
I mean, when we talk about the mutation, my first thought is that it comes from the radiation. We are all the time exposed to the radiation, the background radiation of the nature. And also sometimes we do the X-ray check, especially for the CT scan. We will receive a lot of radiation in a short time. So people talking about the dosage, and they say it's okay. You just received some dosage like 1 year in the natural environment if you take an X-ray. But I wonder if you receive such a high dose in a short time, relatively high dose in a short time, does it? I think it's more severe than you receiving low dose, like the background radiation for a long period of time, because you have more time to repair your mutation. But if you receive intensive dose, not that intensive, I mean, like X-ray scan or CT scan, is that more severe and causing more DNA damage than the equal amount of dosage that you receive in a long time? That's my question. Thank you.
Okay. Yeah. Thank you. Thank you for your question. I mean, the question that you ask is about acute radiation and chronic radiation. So in my topic so far, when you heard is about acute radiation. So we only gave them one dose of radiation at only one time, with different dose of the radiation and observing the effect. They also have a study working on chronic radiation, which they are giving the radiation in a kind of a rate, like they are exposed to certain level of very low level of radiation to mimic some occupational exposure. Like for example, for people who is working in the hospital, dealing with the radiation medicine or working with a nuclear power plant, they are exposed to chronic radiation. That is definitely different, but at least from what we found, like in our supplementary as well, we found that even with very low dose of radiation, it may damage the sperm. So the mature sperm, when they are finish the meiosis, that when they becomes spermatized, they are unable to repair any kind of the damage. When no matter is it a low dose or a high dose of irradiation, even in our supplementaries figure 7, we also observed in the nature environment, in the physiological condition, even they do not get any radiation, they still can having double strand break in their sperm. That is can coming from different other reason. But no matter chronic or acute radiation, low dose or high dose, mature sperm cannot repair any of this damage, and then it can all leading to a certain kind of transgenerational effect. Its only difference is how many damage they can induce in a single sperm. Whether in our case we use very high dose, they induce so many double strand breaks, and when there are so many breaks, they can making a lot of more mistakes or more misfusion between the break points. But when there is only one or only two double strand breaks, maybe they can repair more accurate and they make less mistake. But this is all depends on the, I think, depends on the chance. But another thing about the chronic radiation is whether it will affect early stage of spermatogenesis. And that is another question. I mean, I haven't studied the chronic radiation, but I know some people from other lab who has done some study on the chronic radiation exposure on early spermatogenesis. And that one shows it's going to lower the sperm quantity, so they will generate less sperm cell. So I do think irradiation, no matter it's chronic or acute, it will affect the sperm. I mean, from different way, but the mechanism with chronic radiation on the early spermatogenesis, so far I don't think it's very clearly understand yet. Yeah, if I answer the question.
Thank you very much. So it seems they have to implement some shielding for the sperm bank.
Mm-hmm. Sorry?
I mean, you mentioned that for mature sperm, it's very sensitive to radiation.
Yes.
So I'm
yes.
wondering if they need to make some shielding structure for the sperm.
Yes, definitely. We do think that like in the sperm bank, they should be specially protected.
Okay, thank you very much.
Yeah, no problem.
Yeah, thank you so much. The Victoria asked in the chat one last question. If this study, if you or somebody is planning to move this to other models, and check if the mechanism is the same, but you already kind of hinted that this is a very conserved mechanism. So
Yes, we are also thinking about using mice or using other model organism, but it will be our next step. But so far, by analyzing the data side, looks like it's a conserved mechanism. Yeah.
yeah, thank you so much for these questions, and thank you so much for answering so many of our questions and for giving this talk. This is really interesting and groundbreaking research, and I hope the best for the future, and we are very curious to learn what comes
Thank
next
you.
out of your lab. Peter, did you have something you wanted to say?
Yeah, I don't really have any good questions, and if I have questions, I probably have enough of them to fill the rest of this week. So if we get started on that, but the way I look at this is that DNA is kind of hardware programming in a way. Epigenetics is ways that you can affect how the DNA and the hardware actually works. And then on top of that, you also have the software, how we program the brain, so all of these things, how that can be affected. And where I come from a background where I've been playing around with elite sports and extreme things in a whole lot of areas, both physical and how we affect our brain and how we can use that. And how that can be affected and our understanding of that, it's extremely poor. And these are areas that I think has massive potential. And I think it's very underappreciated because it's talk about like Elon Musk's Neuralink and all of that. But if we ignore that and look at what we can influence by food, training, environment, and what to avoid and what to do, and also how we can influence our brain programming by completely different means, it has potential that is beyond what most people realize. And I've been playing around with it for most of my life. And what can be done is absolutely amazing. And it's very poorly understood as far as I can see. And I think what you're working with, I don't know if this what I just said is things that you're thinking about or touching, but I think the area is extremely interesting. And if what I said is of any interest, tell me, because I probably have ideas from a different angle than what research you're doing, because I'm not coming from the same aspect. I'm looking at what has been working and so forth, but it links into what you're looking at. So I'm very curious, even though I can only ask dumb questions when it comes to the specifics of what you're doing. So
No, I
anyway,
think you mentioned was a brilliant question. I have never touched that field yet, but was very fascinating.
yeah, it's a lot of things that people believe are impossible are possible. And it's so much talk about people having talent, but the human body, and I think it has to do with epigenetics too, because you can trigger the body to respond to training and a whole lot of things that the body can suddenly respond very different than it did previously. So what the body can develop into when you push it and when you train it in sports or any other area is quite amazing, and it's very hard to predict before it's actually tested. So the idea that people are born with talent, yes, but you do not know with the individual before, and many more individuals respond to certain inputs and training, the same as you said there. But and if you have 2 seconds, I have one specific question. If you have.
Yeah, sure.
My
Definitely.
specific question, and you may have answered it, I'm going to re-listen to this. What did you say about, because it has always been curious to me, like the bombs, the nuclear bombs in Japan, that the cities Nagasaki and Hiroshima are rebuilt and people seem to live there very healthily. So the damage that was caused, that seems to have been very temporary, even though it's a sensitive subject. So do you know anything, and that you could enlighten me, that it doesn't seem that it's caused damage?
Yeah, I think that's what I found in this paper as well. I mean, that is because the time range when can leading to this transgenerational effect is very short. So in this kind of accident, no matter in the Second World War, the atomic bomb, or during the Chernobyl accident, this is like a disaster, is a big event. I mean, people get radiation at this event, and until they decide to giving birth to their next generation, I think it's normally not happened at the same time. And normally that can leading to this transgenerational effect is when you're exposed to the radiation, and then within a very short period, I think this is based on my speculation, should be within 1 month, then you get conception. Then it can lead to the transgenerational effect. We're missing the indication of the transgenerational effect because the people who got radiation during this event do not having conception within 1 week or 1 month during this period. So their sperm already repair the damage, because immature sperm or early stage of the spermatogenesis, they can repair the damage. That's why we do not have any evidence of transgenerational effect from this kind of accidents. But I think my study will be more meaningful for people who are regularly exposed to the radiation. For example, the doctor or doctor working with radiation, or the worker working in the nuclear power plant, that is more meaningful for them rather than for the accidents. Yeah.
Very, very interesting, because there's been a thing that I've kind of thought about why that was and why it is, and it's the first time I've heard someone that have that have looked at it. So thank you very much.
You're welcome.
Yeah, thank you so much for that question, because the resilience and the training and the environment question, I think, and then health and modify what you're talented in, or human health for aging. I think that's also something you are interested in your lab, maybe in the future,
Yes.
right? As far,
Yes.
yeah. So it would be interesting also to learn about that part of your of your work one day. So whenever you want to come back, you're always invited.
Yeah, sure. I mean, once I got new new data that want to share with you, I mean, nice stories if I had, then I will definitely will come back.
Well,
Just
thank you.
one
Yeah,
comment
go
there.
ahead.
Yeah, just one comment. One of the things that I find so sad when in what is talked about is that the people are referencing talent, which kind of tells a whole lot of people that, oh, I don't believe I have talent. It's other people that have it. So I don't even try, which for sports may be one thing, but it's so many other things that may have massive health effects that people are like, yes, I just am stuck with this. And I believe that there is so much that people are not even trying that could have massive effects that are not that hard. And I find that really, really sad. So this was a side note, but I think it's something that is very, very important, combined with the research and so forth. But it's also what people believe is possible, and also have a path to actually do things. So anyway, thank you. Sorry for maybe going off on tangents here,
No.
but I find it
Thank
fascinating.
you so much.
Yeah, thank you. I think it's a very important point, Peter, in general, because back in time when I was in school, the physics teacher told my mother in Germany, yeah, she's good for a girl, but she's still a girl. and um, and all these things, I think they suggest you talent, or that this is way too hard for you to understand, and then your perception of the whole subject is maybe different. And then also, yeah, what people can still do in aging, or when you're older. I think it's changing a little bit with research that's just coming out. But yeah, I really agree that it's sad, and that, you know, the more research we will get around this, will hopefully change that mindset. And, you know, epigenetic and histone modulations are also important in the brain for memory and so on. So I think it's a very exciting field. So yeah, as I said, I'm curious to see what will come out of your lab in the future. Congratulations for being
Thank
persistent.
you. Next time I will try again.
No, it was wonderful discussion. We really appreciate it, and yeah, good luck for everything. I hope you get a lot of grants. You probably have
Thank
already.
you.
Thank you.
Thank you both, Katerina, Yao, and everybody else. I really enjoy it.
Yeah, thank you, LT, for your questions, and yeah,
Thank you.
thanks everyone for coming. It's always so much more interesting discussion if different, people from different backgrounds come
I have
together and discuss it.
a question.
Hey, Parth.
Hi, Katrina. How are you? So I joined a bit late, but I got the gist of it. I'm actually downloaded the paper. Thank you for doing this room and for the talk and for being here. So I came in when I think Katrina or somebody was asking about doing these studies in other model systems. Aren't there already many instances in which humans have been affected and their subsequent offspring, several generations, people are still seeing effects? For example, I'm from India, and there was a gas tragedy in 1984 in the city of Bhopal in Madhya Pradesh, where Union Carbide, I think it belongs to DuPont company, I could be wrong, but Union Carbide's a pesticide plant there was an accident. It resulted in the death of 3,000 people in a single day. And even today, generations after that are still feeling the effects because children are still born with brain defects, etc., etc. This is definitely inherited. So maybe it could be a good. It could be good is a wrong word because it's a bad thing that happened there, but that offers an opportunity to examine those people and do a genetic, genome sequencing and find out where the mutations have been carried over from their parents. I can back channel you the articles related to that incident.
Oh yeah, that's definitely. I haven't really looked at this accident, but currently we are also working with some other kind of source of the DNA damage to the father, but I'm not sure if that will be a similar effect. But I mean, I don't know whether in that accident was also the similar mechanism because different kind of the damage or stress happen in the parental generation already known they can be transgenerationally inherited mostly from the mother. From the father is mainly from the DNA damage. Not sure if that is a similar thing that in this accident, but if so, it will definitely worth to look at the genetic data from the offspring to see if they have shown the similar structure change, like what I see in the C. elegans in the worm. If that's so, I mean that would be a very interesting project to continue. I mean, our work also provides some potential therapeutic approach. For example, we can use the histone H1 or heterochromatin inhibitor treatment to see whether it can prevent the offspring effect. I think maybe that would be future work to look forward.
Yeah, thank you. I'm trying to put some links in the chat section. Yeah, thank you.
Oh yeah, I will have a look.
Yeah, and the good thing about recording this on Clubhouse is that the chat, when you click on the recording on the replay, the chat links will still be there, and the chat and also all the links and everything will stay active, that you can just click on them. So yeah, if anyone wants to check out resources afterwards again, you will see them in the replay. So thank you, Parth, for that interesting suggestion. Okay, great. So thank you everyone again, and of course a
Yes,
special
Catherine, I'll just add couple more points. So it was one of the most horrible disasters that happened because the Union Carbide company, which is based in the US, it was a subsidiary in India. They immediately tried to dissociate themselves from that accident, but they are the ones who caused it. So there was a large scale public protest, and the court proceedings went on. I think the company's main head, Warren Anderson, I think that's his name, he immediately fled the country, and it took a long court dealing to settle the claims. And even then they were given, I think, a paltry $400+ million, which is nothing compared to the amount of damages they caused. So it's still, the victims have not been given justice. So it's something that people should know. And as in a significant way, the then Indian government was also responsible for it, because they did not take adequate measures to bring justice or give due compensation to the victims. Thank you.
Yeah, thank you for sharing the story. This is a really sad, real world example of humanity not doing a good job protecting humanity. And I hope that people get the compensation they should get. So again, thank you, Yao, for your presentation. And I'm really I thank you also for mentioning that your in the future therapeutics will come out of this work. That is very promising, and thank you so much for doing this work and for staying on top of it and being persistent again. Thank you.
Thank you for the invitation. It was really a great experience. I mean, I have never done that. It was really, really fun.
Oh,
Thank
that's
you.
so great. We are always happy to hear when the speakers that we invite are also happy. That's the best combination. So great, and enjoy the rest of your week. And
You too.
everyone, thank you so much. And we have our next room on Thursday at 4 p.m. EST with Professor Perrybone and also the group, talking about dual polarity voltage neuroimaging. It's like a new way of doing precise neuroimaging technique with results, which will be really interesting, and the group will be coming. So, and again, this was a wonderful discussion. Thank you, everyone, and I'll close the room in 3, 2, 1. Bye, everyone. Thank you.
Bye.